Happy Sunday, everyone. Welcome back to Life of Pri. You might be able to tell by the background, but I am on campus and heading into lab. I have a DNA extraction to do today, and it's going to be a bit of a tight squeeze before choir practice, but I think I can do it. Actually, I have to be able to do it in time. So, let's go.
Okay, so remember that DNA extraction buffer I made yesterday? Well, today I'm going to use it, and I could basically do the DNA extraction in my sleep now. After 4 years of doing it, I have it down. Anyway, the first step is to add 400 microliters of the DNA extraction buffer to each of the wells. From there, we put it into the oven for 30 minutes at 65 degrees. I then have to move to the fume hood because of this guy here, ammonium acetate, which is corrosive, so don't spill it. We then put it in the fridge for 15 minutes and let the reaction occur, and after the 15 minutes is up, we centrifuge it for another 15 minutes. While they're in the centrifuge, we then get new plates and add 360 microliters of isopropanol to each well. Now, isopropanol is an alcohol, and so it's super slippery. FYI, anything that ends in OL is an alcohol. Now that the plates are done centrifuging, it is time to take 600 microliters from each of these green wells that have the plant DNA and add them to the wells that we just filled with isopropanol. And this will actually make the DNA precipitate, and it's visible to the naked eye for a period of time. And from here, we are going to centrifuge it again and bring all the DNA down into a pellet. While the plates are centrifuging is a great time to clean up your messes. So during this centrifuge, I'm going to take the old plate mats and put them into a 10% bleach solution overnight, so they'll be nice and clean and decontaminated for the next round of extractions. The next round of centrifuging is done, and now we actually decant the supernatant into the waste bucket and tip them on their sides for a minute to let them drain. After that, I then add 400 microliters of 70% ethanol to clean the DNA pellet. And guess what? We centrifuge it again. While it's centrifuging, I'm going to discard my waste, which has ammonium acetate, into the hazard waste bucket. And we are almost done. The final step after the last centrifuge is to once again decant the supernatant and to let the tubes dry upside down overnight to remove any leftover ethanol. And tomorrow I will add DI water to the tubes to let the DNA dissolve into that. And that's what we use when we do our PCRs and PACEs. So there you have it, a DNA extraction.
Finished the DNA extraction with time to spare, so woohoo, time for lunch before choir practice. During the centrifuge interludes during the DNA extraction, I got to video chat my friend Jacob from undergrad, and so that was a lot of fun. I hope to see him when I go home for my sister's graduation. So we'll see. Fingers crossed. He's one of my best friends from undergrad, and it was just nice to chat and catch up and see what was going on in life. Anyway, from here, choir practice, mass, maybe downtown for boba. We'll see, and then maybe lab again. Stay tuned. Hey everyone, so I'm here with Patrick and Josh, and we ended up at Baskin Robbins. You can see the backwards BB up there and all the flavors of ice cream. We are getting milkshakes, and we all got mediums, which apparently equals 3 scoops. So we're gonna have a sugar high later, but yum.
And when I say yum, I basically mean be jealous. These were delicious.
Wow, what a day. So obviously you can tell I'm back at my apartment, but not for long because I'm heading into lab to finish up that PCR. I'm telling myself to do it now because then it'll be ready for tomorrow and I can analyze it, and I'll be one step closer to being done with everything that needs to be done before going home. So yeah, I can do it. Let's go. So this is pretty neat. This PCR uses a master mix called Fire Plant Master Mix, and it has a green dye in it. And each of these wells is actually a different combination of primers. On the left side, I start with primer 3 and reverse 1, and then over here on the far end is primer 5 and reverse 1 or reverse 10. I know the order. So yeah, I'm going to test these out at 58 degrees and see which of these combinations works the best. Fingers crossed one of them works. Success once again. Have the PCR running, and I am done for today. I don't think there's any more like work I need to do besides keep, you know, always more can be done on my paper. But I think I'm going to go home, maybe go on a run. That usually helps with stress. Go on a run, make dinner eventually, and just let my brain shut down for the rest of the night, because on Monday and Tuesday, we got to get back at it. Anyway, I hope you all had a wonderful weekend. Hopefully not as busy as mine, or if it was busy, more fun at least. And I will see you all tomorrow.